Good morning, sunshines. We're going to go through some work on genetics today. Even though we're not at school together, we missed this really important unit of biology, so I'm hoping we can at least review some of the stuff that you learned in middle school. This also reviews a little bit of the work that you did last week in your web quest, your intro to genetics, and some talk about Gregor Mendel and his pea plants. All right, first up, our vocab. We have 7 terms that we're going to be using in this week's lesson, and I just wanted to make sure that you have seen these words and that you have the definitions written out for you so you can refer back to them anytime you need to. Genes, traits, allele, genotype, phenotype, heterozygous, and homozygous. And we'll go through the definitions as we get to each slide. First, it's the work of Gregor Mendel. You can see in this picture some of the traits that we talked about with Miss Densmore when she was with us for that week before schools got closed. And we talked about a widow's peak hairline. Oops, sorry about that. We talked about a widow's peak hairline versus a straight hairline. We talked about how some people have a hitchhiker's thumb and others have a straight thumb. We talked about how some people can roll their tongues and some people can't. And then we talked about attached earlobes and unattached earlobes. And each of these things are traits or physical characteristics that you have. But your traits are determined by genes, the information written on your DNA, the orders of the A's, T's, C's, and G's, those nucleotides that we learned about. When the order changes, the traits change or the physical expression changes. So this lady up in the top left-hand corner, she has a widow's peak hairline, where the other lady has a straight hairline. There's one gene for that, that's the hairline gene, but there are 2 different versions of the gene. One version makes a straight hairline and one version makes a widow's peak hairline. And those different versions are called alleles. And that's what we're going to work on today, trying to understand what an allele is and how we can write those to represent individuals. Gregor Mendel is the first one who discovered this, and he did his work back in the 1850s. And it seems like common knowledge to us now when we talk about like dominant traits and recessive traits. You guys have learned about that before, and a lot of people know about it, but in the 1850s it wasn't common knowledge. Gregor Mendel worked in a garden at a monastery in Austria, and he started working with pea plants. And he kind of got lucky that he worked with pea plants. They have very simple traits, where other plants can have much more complicated traits, and Mendel might not have reached the conclusions that he did, but he was working with pea plants. It helps him that the pea plants, are self-pollinating. That means that one plant will produce both egg and sperm, and it will fertilize itself in order to make offspring. So that one plant acts as both the mother and the father. And because they are self-pollinating, these plants are true breeds. That means there's no difference, no variation from parent to offspring. They're always going to be the same. So if mom or the parent has one particular trait, then the offspring has to have that trait also. They're true breeding. So Mendel's pea plants were self-pollinating and true breeding. And when Mendel looked at his pea plants, he studied them very intensely, and he realized that they had lots of different characteristics: seed shape, seed color, seed coat color, pod shape, pod color, flower position, and plant height. And here's what each of those things looked at, what Mendel observed. For seeds, he saw that there were 2 different seed colors, yellow or green. He saw that the seed coat could be either gray or white. The flower could either be white or purple. The pod could either be smooth, this says full, but sometimes you'll see it called smooth, or constricted. The pod could be yellow or green. The plant stem could either be tall or short, and then the flower position on the stem could either be axial or terminal. Axial or terminal. Axial means that it comes off of the stem, and terminal means it's at the end of the stem. So these were the 7 characteristics that Mendel looked at when he was studying his pea plants. And he did something. He had them do something called cross-pollinating. Remember before we said that pea plants are self-pollinating, where one pea plant acts as both mother and father. Well, Mendel decided to cross-pollinate. He took the sperm, the pollen, from one pea plant, and he carried it over to a second pea plant and dropped it off so it would fertilize the egg of the second pea plant. That's cross-pollination. The egg and the sperm will come from different plants. And we have these words to review from last week. We have a parent generation and an F1 generation. Parent is written as P, that's the parent generation, and F1, first filial, Latin for sons and daughters, so the kids. So we've got our parents and our kids. Mendel thought if we've got a plant with a round seed and another plant with a wrinkled seed, what would we get over here in our offspring? And he thought, well, maybe those plants would be just slightly wrinkled, that these two traits would blend together. That was what he predicted what would happen. So if we have gray and white seed coat colors, the blending over here would be more like a very light gray seed coat color. And if we had a tall and a short plant that Mendel cross-pollinated, he thought that their height might be like medium, blending of the characteristics. So Mendel did his experiment. He grew his pea plants, or cross-pollinated his pea plants, got the seeds and planted them and made the offspring grow. And he found out that all of the offspring expressed one of the traits of the parent, but the other parent trait seemed to be gone. None of the offspring were wrinkled. None of the offspring had green seeds. None of the offspring had white seed coats. None of the offspring had constricted pod shapes, or yellow pods, or terminal flowers, or short stems. All of those traits just seemed to disappear. They didn't blend like he thought they were going to. One of them disappeared and the other trait stuck around for the first filial, the F1 generation, the kids. Mendel wasn't expecting that at all. It caught him by surprise. So he thought, okay, what if I take those plants, the F1 plants, the kids, and we let them self-pollinate? Remember, self-pollinate means that one plant acts as both mom and dad, provides both the egg and the sperm. And he wants to see if that other allele or form of a gene has disappeared. What he gets now is the F2 generation. This would be like the grandkids. So we have the parents, we have the F1 that were the kids, then he lets the kids self-pollinate and they make the grandkids, or the F2 generation. And his results, he saw that that other trait came back in a quarter of the plants. The other 3 quarters still had the dominant trait. So if we go back to this one, by the time we got to the F2 generation, 75% of them would be round, and 25% of them now had this other trait that seemed to have disappeared, the wrinkled trait. Because of these experiments, Mendel came up with 4 conclusions. At the end of this video, you'll have some questions to answer on Schoology, and some of the questions are about Mendel's conclusions. So he had 4 conclusions. Here's the first one. Biological inheritance is determined by factors that are passed from one generation to the next. We know now, or now we call those factors genes. Things that are passed from one generation to the next, and genes are written on your DNA. Then he gave us this word alleles. Alleles are the different forms of a gene. So for plant height, that was the gene. There was a spot on the chromosome for a height gene, and there were two different forms, tall and short. Mendel's second conclusion was the principle of dominance, that one of those alleles could be dominant over the other allele. That means it hides the other allele. And we decided to write that with a capital letter to show that it's dominant. The other allele is recessive, and it's hidden by the dominant allele. And we pick our letter based upon the dominant. So for plant height, our two options were tall and short. Tall is dominant, so we're going to use the letter T to write tall, and we'll use a lowercase t to write short. Some people have a hard time with this. For short, they want to write the letter S and write it lowercase, but we can't have a lowercase s because different letters represent different genes. Sorry for my dog, he's excited with his chew toy. All right, Mendel's third conclusion that he came up with is something called the law of segregation, and we've already learned this one. We learned that you have two copies of a chromosome, one that you got from your mom and one that you got from your dad. They belong to you now, but when you're making your egg or your sperm, you have to split them up. You cannot give both copies of your gene to your offspring. They have to get the other copy from the other parent. So that's Mendel's law of segregation, that the two alleles, the two forms of the gene, segregate or separate during meiosis. And then Mendel's fourth conclusion that we're not going to spend a lot of time on is the law of independent assortment. And that says that the genes for flower color and plant height will be passed down to their offspring separately. Basically, if a plant has purple flowers and it's tall, that doesn't mean its offspring have to have purple flowers and be tall. They could have white flowers and be tall, or purple flowers and be short. So now we've gone through some of our terms and some examples of Mendel's experiments. We're going to do some Punnett squares on our own. And Punnett squares show probability. Probability is the chance that something will happen. Just like when you flip a coin, there's a chance, a 50% chance that that coin will land heads up and a 50% chance that that coin will land tails up. Probability is just like that. And we can write our probability as a fraction, as a ratio, or we can even write it as a percentage. Any of those are appropriate. Some words we need to know to do our Punnett squares: heterozygous, homozygous, phenotype, and genotype. Hetero means different. And in our case, we're going to talk about two different alleles. Same gene. This shows that we're talking about one gene, the gene for plant height. But this organism has, or this individual has, a dominant version and a recessive version of that plant height gene. Homozygous means same. So that means this individual, one gene, plant height, has to have two copies of the plant height gene, and they could either have two big copies or they could have two little copies, two recessive copies of the plant height gene. As long as the letters are the same capitalization or lowercase, then that counts as homozygous. Phenotype is the physical characteristic, or what you can see. When you look at a plant, you don't see big T, big T. What you see is a tall plant. And genotype is the genetic makeup, or what you don't see, like big T, little t, little t, little t, or big T, big T. You can't see those things when you look at the plant. All right, let's set up a Punnett square really quick so we make sure we remember from middle school what to do. First, you've got one parent that you're going to write across the top. And remember, each parent can only give one copy to their offspring. So this big R, little r, we're going to split them up. One goes over here into this egg, and one goes over here into this egg because each parent can only give one copy to their offspring. The second parent gets written down the side. So this parent puts one R in the sperm and one R in the sperm. And then we're going to fill out our Punnett square to see what would happen if this egg and sperm came together. The offspring would have two dominant alleles. If this egg and sperm came together, the offspring would get one dominant and one recessive allele. We always write the dominant letter first. If this egg and this sperm came together, the offspring gets one dominant and one recessive allele. And if this egg and this sperm come together, the offspring gets two recessive alleles. We'll do a practice one together here. A green pea plant, big G, little g, is crossed with another green pea plant, big G, little g. Predict the genotype and phenotype probabilities of the offspring. We'll only take one parent and write them across the top. Remember, this is like showing that one allele or one copy of the gene can go in each egg. So we separate the big G and the little g. The other parent gets written down the side, and we show that the letters get separated in the sperm. If these two come together, we get big G, big G. Down here, we get big G, little g. Over here, we get big G, little g, and little g, little g. Predict the genotype and phenotype probabilities of the offspring. Genotypes. Those are the letters, what we can't see when we look at the plant. So these are our possibilities for our offspring. This does not mean we have 4 offspring. It just means this is our possibility. And we have a 1/4 possibility that the plant will be big G, big G. That's the same thing as saying a 25% chance. Then, big G, little g. We have a 2 out of 4 chance, or a 50% chance that the offspring will be big G, little g. And little g, there's one of those for another 25% chance. That's for our genotypes. Phenotypes. These are what you see when you look at the plant. These 3, change my color here. These 3 plants, or these 3 boxes in the Punnett square, all have the dominant letter, the capital G. So all of those, when you look at the plant, they will appear green. It doesn't matter that 2 of them have a little g. That little g disappears. It's hidden by the big G allele. And then we have one plant that is little g, and the recessive version of this gene is yellow. And that's 1 out of 4, or 25%. Nothing in our problem told us yellow. We didn't know that it was going to be yellow at all. The way I knew it was going to be yellow was looking back up here at Mendel's pods and seeing that yellow was the other version of the gene. All right, let's do another practice problem. A homozygous tall plant is crossed with a short pea plant. Predict the genotype and phenotype of the offspring. Now this one tells us homozygous. Homozygous means same. So they have to be T's, but they have to both be either big T or little t. And this one's tall, which is dominant. So we get 2 big T's. And we'll split them up into 2 eggs. And then we have a short pea plant. That's little t, little t. We'll put those into the sperm. Fill out your Punnett square. And predict the genotype and phenotype probabilities of the offspring. Genotype, only one is possible. 100% of the offspring will be big T, little t. And for the phenotype, again, only one thing is possible. 100% of the offspring will be tall because each of these boxes has a big T. So tall shows up. This is the experiment Mendel did from his parent generation to the F1 generation. He saw that he had one tall parent and one short parent, and he crossed them, and 100% of his offspring ended up tall. All the short alleles seemed to disappear. According to this Punnett square, though, we see it didn't disappear. It was just hidden by the dominant gene. All right, this one's a little trickier. It does not give us the genotypes. A heterozygous red flowered plant is crossed with a white flowered plant. We have to remember what heterozygous means, and it means different. So since it means different, we know we have to have one big letter and one little letter. If it has a big letter and a little letter, that means this plant has to show up as the dominant version. So we've got to decide, is red dominant or is white dominant? And we would say that red is dominant because in the heterozygous one with one big letter and one little letter, this plant shows up red. Since we're talking about one gene, flower color, to write the genotype for the white flower, we still have to use the letter R. A lot of people want to use W's because it's a white flower plant, but you can't do that. It's one gene, flower color, and the versions are red and white, but we use the letter that goes with the dominant version. So big R, and then we have to use little r. Big R, little r is a red flowered plant, and little r would be a white plant. We split our letters up. Predict the genotype and phenotype probabilities of the offspring, and we get big R, little r would be 2 out of 4 or 50%. And little r would be 2 out of 4 or 50%. So we expect about 50% of the offspring to be big R, little r, and 50% of the offspring to be little r. For phenotypes, big R, little r would show up as red, and little r would show up as white. Remember, these Punnett squares don't show you that they're going to have 4 kids and 2 of the kids are going to have red flowers and 2 of the kids are going to have white flowers. It's just a chance that every single time these 2 flowers reproduce, or these 2 plants reproduce and have offspring, there is a 50% chance that their offspring will have red flowers and a 50% chance that their offspring will have white flowers. Here are the 5 problems that I'm putting on Schoology for you to practice, and then I have a couple questions about Mendel's conclusions and some vocab words for you to answer. You're going to have to look back at your definitions, heterozygous and homozygous, and make sure that you know what those are, and you might also have to look back at this chart to see what is a dominant trait and what's a recessive trait, or what the other versions are. I'll make sure you have access to that in your problems. If you have any questions, reach out, let me know. I'll help you as best I can. I miss you guys. Be good, and I'll talk to you soon.